We were looking at the effects of tocopheryl acetate on FLP1 expression, growth rate, and feeding behavior of Tetrahymena thermophila. Tetrahymena thermophila is a unicellular eukaryotic ciliate that is popular as a model organism in molecular biology. So it's been proposed that tocopheryl acetate, which is a chemical that's used as a thickening agent in THC-containing vaping products, is associated with vaping-related illness, or EVALI. And EVALI stands for electronic cigarette or vaping product use associated lung injury. And as of December 12th, 2019, has caused 52 deaths in the United States. Tocopheryl acetate has been implicated and linked to EVALI because it has been found in the bronchoalveolar lavage fluid of EVALI patients. And so in the presence of high amounts of tocopherols, phosphatidylcholines undergo a phase change from a gel to a liquid crystalline phase, which reduces the ability of the surfactant to maintain the surface tension necessary for respiration. And so this chemical is found in skin creams and products that can be applied to your skin, but when you inhale this substance is when you really have negative health effects. And so in causing the surfactant to lose its ability to maintain that surface tension, that's the mechanism that we see as possible and how it causes the respiratory dysfunction. FLP1 encodes Flippase, which is used to transport lipids across biogenic membranes. And so we hypothesized that if Tetrahymena thermophila were exposed to tocopheryl acetate, their cell growth, the expression of the FLP1 gene, and their feeding behavior would all decrease. And so with our methods, we use the oligoanalyzer software to design primers to replicate our FLP1 gene. And then our Tetrahymena cultures were maintained in NEF media and then transferred into SPP media for observation. Our experimental cultures were then exposed to a single 72-hour dose of tocopheryl acetate and ethanol. We extracted the total RNA from our Tetrahymena cultures through the use of Qiagen's RNeasy mini kit. And then our cDNA, we were able to produce by reverse transcribing that RNA using the RevertAid reverse transcriptase enzyme. Quantitative PCR was then used to measure the changes in our FLP1 gene expression. And for this, we used the PowerUp Cyber Master Mix. We monitored the expression of BTU1 as well and used that as a positive control. We then counted the cells using hemocytometers to make a growth curve, and we were able to perform the feeding assay by observing the vacuoles after the cells had been incubated with India ink dye.
All right, so on to our results. Figure 1 shows the growth curves that we generated for both the control cells and the cells exposed to tocopheryl acetate. The control cells are indicated with the blue line, and the experimental cells are on the orange line. The error bars show the standard error of each value. We measured cell counts at 24-hour increments from initial exposure of tocopheryl acetate to the experimental cells at 24 hours, at 48 hours, and then lastly at 72 hours. We had an N of 3 for each group at each time interval, and our p-value at 24 hours was 0.982, for 48 hours it was 0.316, and for 72 hours it was 0.594. If you look at figure 2, this is showing the number of food vacuoles per cell after 72 hours of treatment. In red is showing the control cells, and in blue is showing the cells exposed to tocopheryl acetate. On the y-axis is the average number of food vacuoles per cell. And we had an N of 4 for each group. our p-value for the difference between these two was 0.001. If you look at figure 3, this is indicating the fold change in expression of FLP1 in Tetrahymena after 72 hours of treatment. The red bar is showing the control, and the blue bar is showing the cells exposed to tocopheryl acetate. On the y-axis is showing the fold change of expression FLP1, and that's relative to the control group. So the control group had a fold change of expression 1. The tocopheryl acetate had an increase in expression, and that fold change was closer to almost 0.1 or 1.4, excuse me. Now onto our conclusion. The values we got for the cell counts were inconclusive, but overall, Tetrahymena cells that were exposed to tocopheryl acetate grew slower than the control cells. But again, our p-values were all greater than 0.05, which was our alpha value for this experiment. So we were not able to reject our null hypothesis. The feeding assay showed significantly less feeding in the cells exposed to tocopheryl acetate than the cells that were not exposed. This was a significant difference with our p-value being 0.001. And then we were only able to perform one round of qPCR with 2 replicates for each. So we weren't able to find any conclusive results. But after one round of qPCR, we did see an increase in expression of FLP1 after in the cells that were exposed to tocopheryl acetate as compared to the control cells. But again, the sample size was too small to be conclusive. So in conclusion, our hypothesis about the feeding behavior was correct. We did see a decrease in feeding behavior, but the hypotheses concerning cell growth and expression of FLP1 were inconclusive. Our conclusions, as mentioned before, were limited by our small sample size. So future research should seek to expand replicates of cell count assay, the feeding assay, and qPCR, especially the qPCR, as we only had an N of 2 for that experiment. And I'll move so you guys can see our references. And thank you for listening.